Great. Well, thank you very much for the invitation. It's a real pleasure to be here to tell you about parasites. So my lab in Oxford study a variety of different human infective parasites and how they interact with their hosts. Normally they're human hosts. So we study some parasites which infect animals, livestock, but the majority of the parasites that we study affect humans. And we use biochemical methods. We try to understand things at a detailed molecular level to try to understand how these parasites interact with us as part of their ways in which they survive and cause diseases. So the main parasites that I'm going to tell you about today, oh, my thing stopped working. Oh. So the main parasites that I'm going to tell you about today are here on the slide. So on the left-hand side, you can see a load of red blood cells, a field of red blood cells. And those red blood cells in the center of the image, you can see look really sick. And that's because they're filled with malaria parasites. So those little yellow spheres are the parasites that cause malaria, Plasmodium parasites. And these are, as you'll see, intracellular parasites. They have to get inside your cells in order to cause disease. And then that's on the left-hand side. On the right-hand side, you can see in this false-colored image, again, you can see red blood cells. And then in blue, you can see trypanosomes. And they're those kind of wiggly sort of single cells once again, single-celled parasites. But these, instead of living inside those red blood cells, swim around within the blood. And so most of the stories that I'm going to tell you about relate to these two different parasites. So we study other parasites, we study amoebas, we study worms. But today I want to tell you about how these two very different human infective parasites can survive within the human host. So what they get from being inside a human, how they change and adapt to live inside humans, and then how they resist the attack that they receive from the human immune system, because of course the human body doesn't want them there, using up its food, destroying its cells, and replicating. So parasites are in a constant evolutionary battle with humans and other mammals, and have been for millennia. And so not only can we learn a lot about parasites from studying these organisms, we can also learn a lot about the human immune system, because these organisms have been with us for as long as there have been humans, and so our immune systems have evolved and adapted in their context. So we see all sorts of cunning strategies adopted by parasites to avoid being destroyed by our immune systems, but we also see all sorts of cunning strategies which our immune systems use, which have specifically evolved to target parasites. And central in this evolutionary battle is the surface of the parasite, because that's the contact area between the parasite and what's going on around it, the host. And at the bottom of this slide, you can see just a diagram of the trypanosome cell surface. And I'm going to be telling you a bit more about this in a bit more detail as we go on. So what do parasites want from their hosts? Of course, parasites want food and they want shelter. The great advantage of being a parasite is that you can hide away. You know, if you can survive in somebody's blood, it's like being constantly in a sweet shop or a cake shop. You know, you're constantly surrounded by nutrient molecules and oxygen, which that individual or that mammal have taken in through its food. The blood is an incredibly rich place to live. It's full of food, so it's an easy life if you're a parasite that can survive in that environment. You're constantly surrounded by food, and also, of course, the body maintains homeostasis. It maintains constant temperature, it maintains constant osmotic pressure. It does all of the things to maintain the perfect environment for our own cells to flourish and survive. And that means that that constant environment is brilliant for parasites, because they are also not buffeted around by changes in temperature or pressure or different toxins, all sorts of things coming and hitting them. So it's great to live in human blood because it's full of food and it's a great stable environment, a great sheltering place. And so as a result, our blood is full of the molecules and the cells of the immune system. And these are there, of course, to fight back. So on the left there, you have a classic antibody. In the center, you have the molecules of the immune system, the innate immune system, such as complement. And complement is a set of proteins which can bind to and recognize pathogens and kill them in all sorts of different and imaginative ways, such as popping them or recruiting immune cells to destroy them. And so then as a result, on the right are the cells of the immune system, cells like natural killer cells, which will come along and destroy the pathogens, or macrophages, which will gobble up those pathogens. So if these parasites can survive in the human body, that's wonderful. That's for them. They're surrounded by food, they're in a stable environment, and as a result, we have molecules to try to stop them and cells to try to stop them through different aspects of our immune systems. So I'm going to tell you a set of different stories which should come from the work that's been done by the guys in my lab, addressing different aspects of how parasites interact with the human host. And then at the end, I'm going to tell you a story about how we can use this information to try to design vaccines to stop malaria, coming up with trying to target the tricks that these parasites use. So firstly, this is a story about the trypanosome surface. And I showed you briefly a cut through of the trypanosome surface. And the surface of the trypanosome is coated. So what you see in that electron micrograph, which takes up most of that slide, is the surface layer of the trypanosome. And you can see it's much thicker than a normal membrane. And that's because it's coated in this dense layer of a protein molecule called the variant surface glycoprotein. So every single trypanosome cell has about 10 to the 7 of these molecules all across its surface. And this creates a surface coat which protects the trypanosome from being detected. Because the trypanosome can be a shapeshifter, it can switch which of these VSG molecules it coats its surface with. So if our body learns to recognize one of them, it can cut that one off, release it, and it can put a different one on its surface. But how do receptor proteins operate in this context? Because, of course, to get food, the trypanosome is going to want to grab things which are in serum and take them up and use them as a nutrient and consume them. So, for example, transferrin floats around in your blood, and it's the way in which your body transfers iron around. And the trypanosome is going to want to grab that iron and use it. And similarly, heme. Heme is a really useful nutrient for the trypanosome to build all sorts of different things. And it wants to grab heme from your serum. And heme in the serum is in the form of hemoglobin haptoglobin complexes. So how do these receptor proteins function? How do they whiz around and grab material from the trypanosome surface? So this is just a piece of work done by Harriet, a PhD student working with me, and she found out what this hemoglobin haptoglobin receptor from the trypanosome looks like. And it's this blue molecule, this elongated molecule made of these 3 alpha helices in a kind of bundle. And in orange, red, and yellow there, you can see hemoglobin haptoglobin. And this is the nutrient that the trypanosome wants to grab. So you could see how the receptor sits and how it allows that molecule to be grabbed. And then this receptor is going to be linked down at the bottom to the trypanosome surface. Hemoglobin haptoglobin is actually this dumbbell-shaped thing. So what we see here is just one of the ends of the dumbbell bound to the receptor. But actually, the image we see here shows what it would really look like with this dumbbell-shaped molecule with 2 receptors, one binding at each one of these ends. And you can see this kink. There's this kind of kink in the receptor around here. And you can see that this kink allows these 2 receptors to form a cradle in which this hemoglobin haptoglobin can lie. And that basically allows 2 receptors to grab hold of one of these hemoglobin haptoglobin dumbbells. And what this achieves is just to make it much more efficient to take this up. So in the right-hand graph, you can see the blue line shows the uptake of monomeric hemoglobin, haptoglobin into cells. So concentration on the x-axis versus uptake on the y-axis. And you can see that's not taken up very efficiently. But if you have this whole dimeric arrangement, which two receptors can grab hold of one hemoglobin, haptoglobin, you can see it takes it up much more efficiently. And it's really good at grabbing hold of this nutrient and taking it up into the cell. And this is just another example of a trypanosome receptor which can grab nutrients, in this case, transferrin in red. And you can see this looks really different. It's this kind of, the receptor is in these two shades of blue, and then you've got transferrin, this time binding right up at the top. And again, this would be attached to the membrane down at the bottom. And this is how these would lie in that VSG surface. So you can see receptors floating around within this VSG surface, with these allowing them to grab their ligands out of the blood to consume them, allowing them to provide a route by which they can grab these nutrients and hang on to them. But this is a bit of an enigma, because if you remember, I told you that the reason for this layer of these VSG molecules, these grey molecules, the reason for these molecules being on the surface is to protect the trypanosome, so that antibodies can't come along and recognize the surface of the trypanosome. But now it's exposing itself to the immune system by putting these receptors onto it. Well, how does that work? And so that's a question I'll come back to. Before that, just an example of a parasite sheltering. So I've already shown you this microscope image of malaria parasites inside a red blood cell. So whereas the trypanosomes have this amazing cell surface, which allows them to wriggle around inside your blood, malaria parasites hide inside your cells. So in order to so if you get bitten by a mosquito which is carrying malaria parasites, it will inject those parasites into your blood, and they'll go to your liver to begin with. And you won't realize you've got malaria for the first 10 days or so, because the parasite hides in the liver. And after 10 days or so, they pop out of the liver and they start to invade your red blood cells. And then each time they invade the red blood cells, one goes in, and about 2 days later, 10 to 20 come out. And so the number of parasites in your body increases and increases, and you start to develop a fever and get sick. But from the perspective of the parasite, being inside one of your cells is a great place to be, because it hides away from those molecules and cells of the immune system. But surprisingly, when the parasite is inside your red blood cells, it sends some molecules, some parasite-derived molecules, it sends them to the red blood cell surface. So why does it do that? Why does it deliberately put these molecules? So this is an infected red blood cell up here. You can see all the little parasites inside that red blood cell. And then this shows you the surface of that red blood cell. And on the surface of these two families of parasite-derived molecules, that long, thin molecules called PFEMP1 molecules, and then this little orange molecule called a riffin. And I'm going to tell you what these types of molecules both do. So firstly, these PFEMP1 molecules, these long, thin molecules. So why would the parasite put these onto its surface, advertising to the immune system that it's there by sticking them on the surface of the infected red blood cell? Well, the reason is because as your blood flows around your body, it passes through an organ called the spleen. And what you see in this left-hand image is a red blood cell squeezing through a tiny slit in the spleen. So this is a single red blood cell, and you can see it squeezing through a slit which is about a fifth of its own diameter. So it has to really squeeze its way through this tiny slit as it passes around your body. And this is great for the blood, because it's a way in which the body can filter the blood and get rid of anything that shouldn't be there. These red blood cells are incredibly deformable and strong, so they can get through these slits in the spleen and still be really healthy by the time they pop out the other side. But what this means, but when this red blood cell is full of parasites, when it's packed full of these parasites, sometimes up to 20 parasites, it can no longer squeeze through these slits anymore. It can no longer pop its way out through these slits. And so what it does is send these proteins, these PFEMP1 proteins, to its surface, which cause those infected red blood cells to stick to your blood vessel surfaces or your brain, the tiny blood capillaries in the brain, all of these different locations within the brain. Or within the body, or within the placenta of a pregnant woman. And that's what causes severe or cerebral malaria. And the parasite isn't doing it to cause disease, it's doing it to stick it down so it doesn't get sent with circulation through the spleen, so it doesn't get filtered out by the spleen and destroyed. So these proteins are anchors which the parasite puts on the surface of the infected red blood cell to stop that blood cell from being filtered out and destroyed. And we spent a while understanding how 3 different human receptors, shown in blue here, binds to different PFEMP1 domains. So, for example, these are 3 different receptors which are found on all sorts of different tissue and blood vessel surfaces within your body. ICAM, CD36 and EPCR. So they're found on your cells and your tissues, and they're involved in cell signaling. But these PFEMP1 proteins in green and pink and yellow have learned to recognize them, not particularly to modulate them, but just to grab hold of them, to stop that infected red cell being flushed away by the blood and destroyed by the spleen. But this can cause trouble. So, for example, we showed that this is one of those receptors, EPCR, bound to this little red protein here called protein C. And the little red protein is its natural ligand. And what happens when protein C binds to EPCR is that triggers anti-inflammatory processes. And what you'll see through this structural overlay is that the yellow protein, the PFEMP1 protein, binds exactly in the same place as that protein C. So they both bind to EPCR, your human receptor, in a way that overlaps. So in other words, if the malaria parasite uses EPCR to stick down to the endothelium, to tissue surfaces within your body, it's going to stop that ligand from binding to its receptor. And that's what that graph on the right demonstrates, that protein C, the red protein binding, is blocked. And what that's going to do is stop the anti-inflammatory pathways, which is going to lead to inflammation where those malaria parasites stick down. And indeed, malaria parasites which stick to this particular receptor are particularly dangerous. They correlate with severe childhood malaria because you get those children get inflammation in their brains because of malaria parasites sticking down within their brains, both blocking blood flow through the cerebral blood vessels within the brain, but also causing brain inflammation by blocking the function of this receptor. And we also showed that if we have these PFEMP1 proteins which can both bind to EPCR, the receptor I just mentioned, and also a second receptor called ICAM1, if you have PFEMP1 proteins which can bind to both of these are particularly correlated with cerebral malaria. So just EPCR particularly correlates with childhood severe malaria, and both EPCR and ICAM particularly correlates with cerebral malaria in children. So you can see the parasite has evolved something to stop it from being destroyed, and that has inadvertent consequences for the infected human, leading to situations in which that infected human has all sorts of deleterious things happening: brain inflammation, brain swelling, and severe malaria. So we've seen ways in which the parasite puts things onto its surface, whether that is these receptors of the trypanosome, the sleeping sickness parasite, or whether it is the PFEMP1 proteins of the malaria parasites. So placing these proteins onto their surfaces in order to grab food or in order to put them into a position of shelter. But of course, what this means is that these proteins, these parasite proteins, are now exposed to the immune system. They're exposed to the molecules and the cells of the human immune system. And these molecules and cells can recognize these proteins, which shouldn't be there. And they're not naturally present in your body. They can recognize them, and that can target them to try to destroy these pathogens. So how do parasites avoid detection? They must be good at it, because malaria has evolved with us for millennia. It affects hundreds of millions of people every year. These sleeping sickness parasites, so people have been found with these sleeping sickness parasites still swimming around in their blood 20 years after they went to a part of the world where sleeping sickness is transmitted. So, people who've been to visit relatives in Africa and then come back 20 years later without having been to anywhere where there's sleeping sickness, they still have sleeping sickness parasites within their blood asymptomatically. So these parasites are remarkably good at hiding within our bodies. So how on earth do they do that? What tricks do they use? Because they're advertising their presence through these different proteins on their surface. So how do they avoid being detected? One of the ways in which they do this is by having families. So I showed you the transferrin receptor earlier. Well, there's not just one transferrin receptor in the genome of this trypanosome, this sleeping sickness parasite. There's a whole family of transferrin receptors, about 20 of them. But each sleeping sickness parasite cell, each trypanosome cell, will only express one of them and use one of them. And then if the immune system learns to recognize it, it will switch. It will shut down that one that the immune system now recognizes, and it will place a different one on its surface. So it will get rid of the molecule which the immune system has learned to recognize, and it will put a different molecule on its surface, which the immune system now doesn't recognize. And this is a process called antigenic variation. It's changing its surface, it's changing its properties, so it's no longer recognized in the way that it used to be recognized. And we see exactly the same for these PFEMP1 proteins to an even greater extent. So this is an evolutionary tree of PFEMP1 proteins, all of which bind to that EPCR receptor, the anti-inflammatory receptor. And you can see there are hundreds of them. There are hundreds of these PFEMP1 proteins that bind to this same receptor. So if the immune system of the person that this parasite has infected learns to recognize the one of these that it's using, the parasite can then just switch. It can switch, it can get rid of that one, and it can use a different one instead. So it can antigenically diversify. So this use of families of proteins is a great way of antigenically diversifying. And this is something that parasites can do really well. So bacteria have an evolutionary selection pressure to remain really simple as possible so that they can divide quickly. Now, parasites that live inside your blood are constantly surrounded by food. There's no evolutionary pressure on them to remain simple and to remain a small genome that can be replicated quickly. Instead, the evolutionary pressure on these parasites is to survive the attack of the immune system, so they can fill their genomes with all these big protein families which allow them to do antigenic variation, to diversify and to survive. This just shows you mapping even the amino acid residues which directly contact EPCR on all of those different PFEMP1 proteins. Even when you do a sequence logo looking at conservation of those residues, you can see even these residues are variable. Even the ones that directly contact EPCR are variable and plastic. In other words, allowing these processes of antigenic variation. Okay, so what happens when antibodies do learn to recognize these surface proteins? So we've seen that these surface proteins are exposed, and we've seen that families of surface proteins have evolved in these parasites through which they can use antigenic variation to switch which one is on their surface to avoid being recognized by antibodies. But what happens when the antibodies can break through? Antibodies recognize these different proteins and the parasite can't escape. Well, one of the things that could happen that could be bad for the parasite in this case is triggering the complement pathway. So this is really complicated, but I'm just going to simplify it a lot in that, so this is a parasite surface here, and in green you can see this molecule called C3, and this is a component of the complement pathway. And if this molecule C3 gets stuck onto a parasite surface, then it's like a label that's been put on the surface of the parasite. And that label can be recognized by all sorts of different things. So there are loads of downstream things, like opsonization is downstream of this. Opsonization is when antibodies cause, the parasites are caused to clump, and then they get engulfed by macrophages and chomped up. Or on the right, the membrane attack complex, which is another set of complement proteins, which punches holes in the parasite. So this C3 molecule it starts to decorate the surface of a pathogen. It recruits all sorts of stuff from our immune system, which are going to kill that pathogen and destroy it and annihilate it. And there are three ways in which that C3 molecule can get recruited. And you can see one of them is through antibodies. Antibodies can recruit a series of things which cause C3 to be deposited on the pathogen surface. So is this relevant? Well, yes, it is. This shows a mouse experiment done by one of our collaborators, Mark Carrington. And their little fluorescent trypanosomes were put inside these mice, used to infect these mice. And what you can see is that so the y-axis is the number of trypanosomes inside the mice. And you can see that it grows over time, and then it stabilizes. Now this, so the dark blue curve is actually a normal mouse and a normal trypanosome, and you can see the number of trypanosomes grows, and then after about 5 days it starts to fall again. The reason it's falling after 5 or 6 days is because the immune system of that mouse has acquired, through the acquired immune system, it's acquired antibodies which recognize that trypanosome, and now it's able to start killing those trypanosomes. And then I mentioned antigenic variation, and then after 5 days, the trypanosome does antigenic variation and it switches its surface, and those antibodies no longer work, and so then the population of trypanosomes starts to grow again. So you see the trypanosomes are growing, antibodies learn to recognize them, and then the trypanosomes switch their antigen and they learn to grow again. But you notice the purple line, that doesn't happen. Why not? Well, that's a mouse which has had that C3 molecule, the label molecule, deleted from the genome of the mouse. So in other words, this ability of antibodies to destroy the trypanosomes, which is occurring in this dip in the growth curve, that requires C3. It's antibody recruitment of the complement labels, which are then leading to destruction of the trypanosomes. So how does this work? So it turns out that trypanosome receptors have a number of ways of targeting this cascade. They have a receptor which binds to C3, and they have a receptor which recruits factor H. Now, many of your cells recruit factor H, and the purpose of your cells recruiting factor H is that factor H stops C3 from being deposited onto your cells and stops it from your cells from being killed. So the trypanosome puts receptors on its surface which recruit factor H. This is just one little bit of factor H, and this is a model showing the whole factor H in purple and C3 in yellow, and how that factor H then inactivates C3. So they recruit things that stop C3 deposition, and they also put a receptor on their surface which binds to C3. This is a crystal structure of the receptor in blue and the bit of C3 in orange. And this is a cryo-EM structure of the whole of C3, again bound to the receptor. So these parasites place things onto their surface, which binds to these complement components and inactivates them. So in other words, this war continues. So the parasites does antigenic variation to avoid being detected by antibodies, but then the human body learns to generate antibodies which recognize them and recruit complement. And then the trypanosome generates molecules which it puts on its surface, which then inactivate complement in turn. So, and the evolutionary war kind of continues. This is just another example of that, before I talk about how we might use all of this to help us design malaria vaccines. This is a complicated diagram, but all I want you to get from this is that here in orange is a target cell. So let's say that target cell is a malaria-infected red blood cell. And the blue cell is a natural killer cell. And this is a cell from your immune system. And these cells are constantly patrolling your blood and tissue spaces. And every time they bump into another cell in your blood, they make a decision about that cell. They decide, is that cell friend or foe? And they make that decision because they have receptors on their surface, activating receptors in green and inhibitory receptors in purple. And your cells have got lots of molecules within them which bind to those purple inhibitory receptors and trigger inhibitory signaling inside that killer cell. So when one of these killer cells bumps into one of your cells, inhibitory signaling is triggered inside the killer cell, and the killer cell thinks, okay, this is a friend cell, so it then snuffles off and bumps into another cell, and it tests that other cell out, and it keeps snuffling around the body until it bumps into something which activates it, until it bumps into a cell which doesn't trigger inhibitory signaling, but instead triggers activatory signaling through those activating receptors. And then it thinks, oh, this cell shouldn't be here, and it releases a whole set of toxins, like this molecule called perforin, which then makes holes in the target cell. So in other words, you have all of these cells of your immune system which snuffle around your body and bind to and recognize these target cells. And if activating signaling is triggered because it's a foe cell, it's a pathogen cell, not a friend cell, then things are released to kill that cell. So malaria-infected red blood cells are susceptible to this. They're susceptible to destruction, in particular if there are antibodies which recognize those PFE-MP1 proteins, because antibodies can bind to activating receptors. So if the antibody binds to the malaria-infected red cell, the FC region, the constant region of that antibody, can then bind to the activating receptor and trigger that cell to come and destroy the target cell. So that brings us back to this second family of molecules found on the infected red blood cell surface in malaria, these small orange molecules. And nobody had any idea what these did until about 2 or 3 years ago, but we knew that they must be doing something important, because a malaria parasite would not expose a molecule to the immune system if it were not giving it a selection advantage, if it were not helping it to survive, because there's a risk involved in exposing itself to the immune system. So these riffins, they're placed on the cell surface, and there must be a reason for that. So what is the reason? Well, what Tom in the lab showed is that these riffins bind to inhibitory immune receptors. So in blue, you have one of those inhibitory receptors, and the riffin in orange is binding to that inhibitory receptor. So these are the receptors that tell the killer cells not to kill when they're triggered to signal. And in yellow and green there, you've got what's called an MHC class 1 molecule. And these are the molecules which are found on your cell surfaces. So when one of these orange and yellow and green molecules binds to an inhibitory receptor, it's going to tell the killer cell not to kill. And we were intrigued by the fact that this riffin binds in exactly the same place to this inhibitory receptor as the molecule that's found across your cells. So we wondered, is it doing the same thing as this riffin molecule? Is this riffin molecule doing the same thing as the molecule that's found on your cells? Is it telling these killer cells not to kill? So we developed this assay with Alex, a PhD student as well. And what he did was, on the top here, you've got a natural killer cell membrane. And then you've got receptors. This time in purple is LILRB1, which is this inhibitory receptor. And then you've got the activating receptors here, bound, for example, through an antibody. So the antibody is recognizing something on this surface, which is recruiting the antibody, which is then triggering the activating receptors. And now the bottom membrane here, think of it as the membrane of the target cell, for example, the membrane of the infected red blood cell. We don't do that because we're using a type of microscopy called turf microscopy, which just looks at a very thin layer of a membrane, and it's much easier to do this with a supported lipid bilayer. So this is actually a supported lipid bilayer on which we can put whatever proteins we want to put. So we've put something that can recruit an antibody, we've put an adhesion receptor, which helps make the interaction, and then we're putting either the MHC or the riffin that I showed you on the previous slide, either the orange molecule or the yellow-green molecule. So what happens when we do this assay? So what you see here is the contact area between the killer cell and the membrane. So where you see a dark image in this IRM image, that's the contact area. Now you can see normally when an NK cell contacts one of those bilayers perforin in yellow gets deposited, and perforin is the molecule that does the killing. So if the NK cell docks onto that bilayer, it's going to activate it, and that NK cell is going to release that perforin, the toxin that kills the cells. If we put the MHC molecule, which was yellow and green in those structure figures, onto that surface, then you can see the perforin molecule no longer gets released, because that's an inhibitory signaling molecule. That's telling this NK cell that this surface is a host cell surface. It shouldn't be activating itself. And we're really excited to see that when we put the riffin on this membrane as well, also we don't get perforin deposition, we don't get killing. Whereas if we have a mutant riffin, which has a single point mutation, which means it doesn't bind to that receptor, once again you get perforin release and killing. So in other words, this NK cell activates in the first line because it's got activating signaling and no inhibitory signaling, but both the human molecule, the MHC, and also this riffin, this parasite molecule, are both able to stop that activation. So in other words, the riffins appear to exist in order to prevent natural killer cells from destroying malaria-infected red blood cells. But our own immune systems have done something very cunning here as well. So normally antibodies, in yellow and blue here, you have what is called a Fab fragment, which is the bit of the antibody which recognizes the pathogen. This shows you one of these riffin molecules in light blue bound to an inhibitory receptor called LILRB1. Sorry, that's called LAIR1 actually. That's LAIR1 inhibitory immune receptor. And riffin, so this is a riffin which recognizes LAIR1. Now, there's a whole family of riffins which recognize LAIR1, and they vary, just as those other families that I told you about vary. So it's very hard to make an antibody which will bind to all of these. So what the immune system has done in people in malaria endemic regions of Africa is the antibodies have grabbed hold of LAIR1. So actually, this whole thing in blue, yellow, and red is an antibody that's found in about 15% of Kenyans. And this antibody has co-opted this immune receptor, this human immune receptor. It's placed it inside its antibody gene, and then it can use it to recognize these riffins. And the job of the riffin is to recognize this immune receptor. So there's no way the riffin can evolve away from recognizing that immune receptor. Otherwise, it wouldn't be able to perform its function anymore. So the human antibody has grabbed hold of that immune receptor and uses it to recognize the riffin in turn. So these extraordinary antibodies have only been found in the last 4 or 5 years, but now increasingly they're being discovered. Antibodies grabbing hold of bits of the other machinery of your cells to use them to recognize parasites. Okay, so just for the last 10 minutes, can we use this information to design better malaria vaccines? So I told you that the malaria parasite in blue has to get inside red blood cells. So the malaria parasite can get inside a red blood cell. There it goes, pop. So the malaria parasite has to get inside your red blood cells in order to replicate and in order to divide and in order to make you sick. Because it's when that malaria-infected red blood cell sticks down within your body that it leads to inflammation and illness. And also the constant infection and lysis of those red blood cells leads to anemia. So it's this invasion process is absolutely essential for the parasite to survive. And it's through this mechanism of red blood cell invasion, destruction, reinvasion, cytoadhesion that malaria makes you sick. So if we can stop this process, we can stop malaria. So it's a great target for malaria vaccines. So this is the form of the malaria parasite which can invade your red blood cells. It's called the merozoite form. It's just a single cell. And it does a number of really cunning things So one of the cunning things that it does is it has these organelles at this tip, and the contents of these little organelles or compartments, these contain all of the machinery that's required for it to get inside a red blood cell. And that machinery is held within these organelles where it's not exposed to the immune system, and it's only released just as the parasite bumps into a red blood cell. And that makes it challenging to block those processes, challenging to design vaccines. Nevertheless, some of our friends discovered this molecule from the malaria parasite called RH5, which is absolutely essential for the malaria parasite to get inside red blood cells. So it binds to a molecule called basogen. And if you increase the concentration on this x-axis of basogen antibodies, you block the invasion of malaria parasites into red blood cells. So you can do this just in tissue culture dishes of blood. So if you have an antibody which stops RH5, the parasite molecule, from binding to basogen, the red blood cell molecule, if you have antibodies which block that interaction, these antibodies stop malaria parasites from getting into red blood cells. Simon Draper's group in Oxford, they do human challenge trials of malaria. In this case, they did an aotus monkey challenge trial. So it's very difficult to come up with model systems to study human infective malaria parasites. And the aotus monkey is one of them. And they do these trials in which they vaccinate and then they inject parasites into the vaccinated aotus. And then the y-axis is untreated. So they monitor parasite burden in those aotus. And when the parasite burden reaches a certain threshold, then they treat them with anti-malarial drugs. So untreated is basically the same as no parasites. You can see the purple line there. Aotus which were vaccinated with placebo, and you can see after 14 days or so they all required treatment because they had all developed parasitemia. And group B, that yellow line at the top, or orangey yellow line, was one of the groups that had been vaccinated with RH5. And you can see that none of those animals required treatment with antimalarials because although they had parasites injected into them directly, they never developed sufficient parasitemia that they required treatment because they showed no sign of illness. So these trials are being done in Oxford on undergraduates as well. So exactly the same protocol is used there. These volunteers have vaccines. They then have parasites injected into them, and then they go for PCR tests every day to check the parasites in their blood. RH5 so far isn't as effective in humans as it was in this animal model, but still the onset of malaria is delayed in that first human trial. And we know why it wasn't quite as effective in humans as in the aotus, and that's because there wasn't sufficient good quality blocking antibody generated in the humans. So what we want to do is use structural biology to design better vaccine immunogens. So rather than sticking the whole of RH5 into these volunteers, we want to stick specific regions of it which just generate the good antibodies. So Kate, another grad student in the lab, was able to see how RH5 in yellow binds to basogen in blue. And then we then started along this kind of pipeline. With Simon, he's injecting these volunteers with RH5 as part of his clinical trials. And we can isolate the B cells. The B cells are the cells which make antibodies in your body. We can isolate the B cells from those people and clone the antibodies from those volunteers. And then we can test those antibodies for the ability to block parasites from getting inside red blood cells, and we can use structural studies to see where they bind to on RH5 and see how they do it. And then we can use that information to design better vaccine immunogens. So each of the squares on this graph is a different monoclonal antibody. And you can see, in this case, the y-axis is the EC30 of the growth inhibitory activity. So the lower it is down on that y-axis, the less antibody you need to block. So the good antibodies are the ones in the red box and the ones in the blue box. Each box represents a competition group. In other words, a group of antibodies which can't bind at the same time, so they bind to overlapping sites. And then on the right there, you can see RH5 in yellow again bound to one of the red antibodies and one of the blue antibodies. So we can see where the good quality antibodies, where the blocking antibodies bind on this structure. And you can see that the vast majority of the antibodies on that graph have no GIA. So if we injected RH5 into you, some of your antibodies, about a quarter of them, would be growth inhibitory, but the majority of them would be useless. So what we want to do is design better vaccines which specifically cause your body to just make the good antibodies. And so this is how we're doing that. So you can see there in yellow RH5 bound to the red antibody which is the best blocking antibody. And what we've done is use protein design to make a small molecule, a small protein molecule, which is this yellow protein here, which is much smaller and simpler than RH5. And this small molecule contains the antibody epitope, the region of RH5 that these good antibodies bind to, stabilize so they fold correctly. So we can just present these little immunogens to the immune system rather than the whole of RH5. So rather than giving a whole of RH5, which generates rubbish antibodies as well as the good ones, this little designed protein specifically elicits or produces just the good stuff. And then that graph shows you growth inhibitory activity, in this case high is good, versus antibody concentration. And you can see the dark blue line is when you use RH5, and all of the other lines are where you use our designed immunogens. And you can see the designed immunogens are all hitting that 50% growth inhibitory activity line with sort of 8 to 10-fold less antibody. In other words, the antibody that's induced by these better quality immunogens is about 8 to 10 times better. So rather than generating all of the rubbish antibodies, we're just making the good ones. And you might ask, well, what about the antibodies that bind around the back of this vaccine immunogen? And that would be a great question. So what we've done is we've designed this immunogen to trimerize. So the epitope regions, the regions which induce the antibodies, are sticking outwards, and the rest of these molecules are packed together inwards, hidden away from the immune system. And then this trimer has been stuck onto a virus-like particle, because virus-like particles are very good at triggering really strong immune responses. So we can use our molecular knowledge of what good antibodies are and how they block to design better vaccine immunogens to specifically cause the body to make just the really good antibodies. And this was used in the COVID vaccine. So not quite this strategy, but a similar structurally related strategy where mutations were put into the spike protein of the COVID virus in order to stabilize in this pre-fusion state, which is the state of the virus that you want to make the good antibodies against. So these structure-guided methods are being used to target, to make good antibodies. So hopefully I've convinced you that studying parasite surfaces is important and fascinating. They do all sorts of cool things. They have all sorts of cunning ways to survive. They teach us new things about the immune system, how complement works, how immune receptors work. And we can also use this information to try to design better vaccines, the vaccines of the future. And yeah, here are some of the people who did all of this work with us, either in my lab or collaborators with us. So yeah, thanks for coming and listening, and do let me know if you have any questions.